Prior to beginning the activity, please be sure to review the faculty information and disclosure statements, as well as the learning objectives. After listening to the activity, complete the post-test by clicking the Earn Credit link in the episode description. Downloadable slides and resources are also available. The following presentation is copyrighted by Medscape. No use, broadcast, or recording of this presentation or any part thereof is permitted without the written authorization of Medscape.
The following presentation is part of a certified educational activity provided by Medscape Education and supported by funding from the U.S. Food and Drug Administration.
Welcome to our program today, Biosimilars, What Primary Care Needs to Know About Biosimilars in Practice. I'm your moderator, Virginia Valentine, with the University of New Mexico Project ECHO. So let's let the faculty introduce themselves.
Hello, I'm Dr. Steve Hanauer at Northwestern University in Chicago. I'm a gastroenterologist and I focus on inflammatory bowel disease.
And my name is Dr. Andy Blauvelt. I'm a dermatologist, and I've worked in clinical trials, and I've done clinical trials with biosimilars, a number of different trials with these drugs.
Welcome. So let's introduce our agenda. In this program, we will discuss the following. Are biosimilars generic drugs? What are biosimilars? Trials and real-world evidence, multiple switches, working with an interprofessional team, biosimilars in primary care, and strategies for talking about biosimilars with your patients. So let's start with, are biosimilars generic drugs?
So when we look at biosimilars, many people call them generic drugs, but it's not technically correct to call them generics. Because when we talk about generic drugs, we're talking about chemical compounds that are exactly the same chemical structure. But biologics are not made through chemistry, they're made through biology. They're actually made through cell lines. So we genetically modify cell lines to produce the antibodies. So the antibodies are made by the cellular factory, if you will, and the antibodies are then secreted from the cell line and collected from the supernatant and purified. And then that is the product. That's the drug product. Now, we can make them very, very, very similar to the original biologic, but they're not exactly the same because the original biologic and the biosimilar are made in different cell lines. So it's the same genetic material that's coding for the antibody, but they're in different cell lines. So the three-dimensional features and protein structures on the outside of the protein, sugar moieties, for example, can be different because the different drugs, the original biologic and the biosimilar, are made in different cell lines. So very, very similar, but not quite exactly the same. So that's why we don't call them generics.
And I would point out also that not all biologics are antibodies. They may be antibody fragments or just large proteins that are used to treat patients' conditions such as erythropoietin.
Exactly. Yeah, I think the point is that the drugs are made through biology and not through chemistry.
So what is a biosimilar?
So biosimilar, again, is a biological product. It can be an antibody, it can be another protein, as Dr. Hanauer mentioned. It's highly similar to the original biologic. So it's patterned after a branded biologic, or we call the originator. And then it's compared in the laboratory side by side, that biosimilar made in a different cell line, patterning after the original. It's tested in vitro in different assays, and they're trying to make it just exactly the same in terms of all the functional assays and the purity and so forth to the original biologic. Now, if we look at the amount of time and effort that it takes to make an original biologic, we see that on the left-hand side here. The preclinical work, the laboratory work for a typical drug is a relatively minor part of the whole drug development process. When the drug is made, yes, it's tested preclinically, it's tested sometimes in animal models, but then it goes into human studies. And we know we have phase one, phase two, phase three studies thereafter. And in particular, the phase three trials for human biologics in this case are very large. They involve hundreds of patients. They take many years to complete and lots of money to complete. And on the other side, when we think of biosimilars, the bulk of the work is done in the laboratory and not in the clinic. All of this testing that I mentioned in the previous slide, the side by side testing, it's extensive. And those are the assays that the biosimilars rely upon the most. Because then when you have a drug that's highly similar from those assays, you don't need much clinical testing. And you see that the pyramid is narrow because previously it was required that only one clinical study really was necessary to show efficacy with the biosimilar. And just as recently as this past year, we're now seeing the FDA saying that just the preclinical studies and not even a clinical efficacy study is required for an approval of a biosimilar. So, very different in terms of the amount of time and effort in the lab, where it's a lot with biosimilars, compared to an original biologic, where a lot of time and effort in the clinic and many years with many patients. And what we get with this is a much cheaper drug development process. So the steps from A to Z, if you will, from making a drug and getting it FDA approved and getting it on the market are much more streamlined with biosimilars. And so the idea here is, or what we call the promise of biosimilars, is to decrease the costs of drug development so that we can increase access to these drugs. So if we can make the costs cheaper, if we can expedite things, perhaps the drugs can be cheaper and more patients can receive these great new 21st century drugs that I call.
And another reason for the lower cost is that the originator needs to be tested in phase three studies for every single indication. Once a biosimilar is approved, it's approved for all of the other indications that the originator has, but it requires the same dose and the same route of administration and the same schedule of administration as the originator for any individual indication.
Now let's talk about some key terms. Now, if you look at the biosimilar literature, there are terms that come up over and over again, and one of them is interchangeability. It's not used as much now, but just we want to make a note of what this term means. This means that when you switch from one biologic drug to, let's say, a biosimilar, say switching from blue to green as shown here, and then switching back, the drugs are considered interchangeable. There's no differences between them. It's seamless. There's no differences in efficacy, safety. So studies that have done this have been actually called interchangeable studies. And then the FDA has given those drugs this particular designation of interchangeability. We're kind of moving away from this, but this is what interchangeability means in the past, is the seamlessness of going back and forth between drugs. Now, another thing that Dr. Hanauer mentioned is the term extrapolation. This is really, really important because, for example, we know with TNF blockers, you have multiple indications. You have rheumatoid arthritis, you have inflammatory bowel diseases, skin diseases, where TNF blockers are used. So the original biologics require testing in each one of those diseases to get the approval for each one of those diseases. So RA studies, psoriasis studies, they all have to be done with the original drugs. But the biosimilar only needed to be done in one disease, for example, rheumatoid arthritis, and then the approval would go to all the other diseases. That's what's called extrapolation. That's the actual term that's been used, so that it got approval for all of the indications of the original biologic without the studies actually being done in each one of those individual diseases.
And currently, Dr. Blava, you've taught me that Nowadays, if the biochemical features are adequate, they don't need to be tested in any of the indications.
Yeah, that was actually a big announcement last year. A big change in the biosimilar field is just all the preclinical work needs to be done now for an approval of a drug, and a clinical study is not even necessary now to get a biosimilar on the market. Big change.
Let's look at some trials.
So, the original study that I'm aware of that looked at biosimilars was actually a study that was funded by the Norwegian government. Many countries outside the US do control drug costs, whereas we do not necessarily control drug costs from a regulatory perspective. In the Norwich study, patients with a variety of immune mediated inflammatory diseases, including rheumatoid arthritis, spondyloarthropathy, psoriatic arthritis, ulcerative colitis, and Crohn's disease, who were receiving infliximab on a maintenance basis, were randomized to continue infliximab at their same dose and schedule, or were switched to an infliximab biosimilar and followed for over 1 year. The aim was to demonstrate non inferiority of switching from infliximab to the biosimilar, and the efficacy was not inferior to continued treatment. Similarly, the safety was observed for both the patients who were continued on the originator infliximab and those switched to a biosimilar. As you can see from the efficacy standpoint, the clinical response rates and remission rates in patients with Crohn's disease, as just one example of these immune diseases, and it applies to all of the other immune diseases as well, was no different in patients who were receiving continued infliximab versus those who were switched to the infliximab biosimilar. There were no differences in the clinical responses, biologic criteria such as C reactive protein, side effects, or in drug levels that were ascertained for both the originator and biosimilar. And along those lines, if you are measuring drug levels, it would be the identical assays for either the originator or the biosimilar.
Let's look at multiple switches.
In this study, which was performed in psoriasis, a biosimilar to adalimumab was used, and patients were either continued on adalimumab or went through multiple switches between the biosimilar and then back to adalimumab and back to the biosimilar and then back to adalimumab, or conversely, started on the biosimilar, switched to adalimumab, and back and forth, and then continued on the biosimilar.
So this is an example, Steve, of a interchangeable study that I mentioned before. So this very simple, biosimilar studies are the first ones were just single switch. You were on an originator and you switched over to a biosim. What happens? This is a much more complex one, and the drug makers at this time were interested in that particular label of interchangeability with this kind of design.
Thank you. And as you can see here, the results of the patients who were remaining on the originator or the biosimilar or underwent multiple switches had the same psoriasis scores than the patients in all of the other groups, demonstrating that you can do multiple switches without losing efficacy or increasing side effects. In other real world studies, not controlled trials, there's good evidence for multiple switches in patients with both Crohn's disease and in ulcerative colitis. You can see that they may have gone through one, two, or even three switches along the way, but there were no differences in their clinical remission rates, biologic remission according to C-reactive protein, or fecal calprotectin. Now, there have also been several safety studies, and these were actually led by Dr. Blauvelt, who should describe what they were.
Yeah, these are actually review papers because the European Union has seen a lot more use of biosimilars over the years. They were much ahead of the US in terms of getting approvals for these drugs and using them in practices throughout the continent. And it's really a rich source of information if you look at the literature. And in these papers, they were extensive reviews of mostly the European data, real world data, showing that there was actually no differences in all the multiple data sources in efficacy or safety or immunogenicity concerns with originator to biosimilar switches. So this is a cumulative evidence.
And this was across different indications, not just one particular disease state.
There's systematic reviews of large amount of data. And then very similarly, this review has occurred of biosimilar to biosimilar switches. So a little bit different. Originator to biosimilar, this one's biosimilar to biosimilar, and it's the same story. There's no changes in efficacy, no changes in safety. These drugs are essentially identical in terms of the effects we see within patients. So it's all good news.
And it's very practical in today's world because we're now seeing some of the third party payers are switching biosimilars every year as they get better pricing. So it's not uncommon for our patients to be switched from one biosimilar to another.
So this is reassuring data.
Absolutely. Lots of real world data as well for many years and many patients.
So let's talk about several clinical scenarios for biosimilar use. The first is for patients who are starting on a new therapy that the doctor has recommended. In this situation, either the reference product or a biosimilar would be equally appropriate. A second situation is when a patient has been on an originator biologic, and then the third party payer makes a deal with a biosimilar company at better pricing, and then the patient is switched from the originator biologic to the biosimilar. Now, in contrast to that, if a patient does not respond to an originator, they should certainly not be switched to another biosimilar comparable to the originator because they're not responding. And likewise, if a patient is on a originator or on a biosimilar and they lose response, in inflammatory bowel disease, we will typically assess why they lost response. If the loss of response was due to just low drug levels, we can increase the dose of the biosimilar or originator. If the patient has developed antibodies to the originator, we should not switch them to a biosimilar because they'll have the same antibodies against that. On the other hand, if the patient is losing response and has adequate drug levels and does not have antibodies, they're not going to respond to a biosimilar and need to be changed or switched to a different mechanism of action.
So let's look at the interprofessional team. Let's first look at the pharmacist's role. The pharmacist to be involved in overseeing the formulary and the IT process, handle financial billing and coding in some institutions, maybe have a different person handling billing and coding in other institutions. They assess literature and develop and identify educational material for patients and healthcare professionals, coordinate with a patient's insurance, including every time there's a switch, and ensure proper dispensing, labeling, and documentation of medications. The physician's role is to serve as the main champions in consistent messaging, whether patients receive an originator biologic or a biosimilar. They're integral to involving patients in the treatment decision making, if possible, and educating them about biosimilars, including explaining why a change is happening, and play an important role in encouraging consistent messaging of biosimilars, including of office staff. The nurse's role is to leverage their close relationships with patients to provide them with meaningful education, often the first healthcare professionals to receive questions and enabling them to alleviate patient concerns and address any issues that arise. They're integral to building confidence in biosimilars. Primary care faculty experiences is having pharmacists provide patient education and facilitate switch to biosimilars, uh, including patients in the decision making process versus just telling them that they're being switched. Sometimes that's going to require a lot of conversation if the payer is involved in making that decision. You want to explain to them why the switch is being done and ensure patients understand the rationale and reassure them, as the doctors mentioned earlier, if your original didn't work, the biosimilar probably won't, and vice versa.
In our practice, once we recognize that the originator can be transitioned to biosimilars, we send a message to all of our patients who are on the originator explaining the biosimilar pathway and how we have accepted it and advocated for individual patients and allow them to address questions back to us through the medical records.
Great. So, you can direct patients to reputable resources, such as things you can get from the FDA and government agencies. There's a list here, different resources to get reputable information. You'll find patients have questions about biosimilars, like how many times will I have to switch? Will it cost less? Are they really as safe as the original? I've been on my current treatment for a long time. Will my disease get worse if I switch to a biosimilar? I'm doing fine on my current treatment. Why should I switch to a biosimilar? How does switching to a biosimilar benefit me? And these are all questions that we've all heard and it can be very disconcerting to patients to have to switch, but it's up to us to reassure them that they are as safe as the original and essentially the same.
Virginia, I wanted to highlight that really the biosimilars are probably good for the overall health system, right? And not so much on the individual level, because we're talking about decreased costs for treating a bulk of patients with expensive medicines with alternatives that are much cheaper. So I think the benefit is more on the healthcare system rather than the benefit on the patient themselves. And I find that the most important thing we can do when talking about biosimilars with patients is reassurance. Reassurance that it's going to be essentially the same efficacy and safety as what they're used to.
Yeah, and they cost less, but they won't cost less to the patient, but like you said, to the system.
And 25 years ago, the major cost of taking care of patients with inflammatory bowel disease was hospitalization and surgeries. Over the past 25 years, since the advent of biosimilars and other advanced therapies, the cost has actually switched to pharmaceutical costs as the major cost, economic costs of the care of our inflammatory diseases, which are Crohn's and ulcerative colitis. I presume the same is with dermatologic conditions, Dr. Blauvelt.
Yeah, absolutely. The cost of treating psoriasis with biologics is very high. Um, it's it's I always say that you're not patients are not going to get those drugs if they don't have the adequate insurance, right? It's all about insurance coverage because the drugs are really overpriced for anyone paying out of pocket.
Now, on the other hand, most of the pharmaceutical companies do have patient assistant plan, and the prescriber should be aware of those and certainly encourage patients to enroll in any of those assistant plans to make certain that they are getting the best pricing for themselves. So make certain you're aware of the pharmaceutical plans and get your patients enrolled.
In my practice, though, one of the caveats is that the pharmaceutical companies will not allow people on Medicare or Medicaid to use their assistance plans.
But they do have end arounds for that. Not always, and I agree that it can be very difficult and not all patients will be covered, but they do have foundations and additional potential support for these individuals, but it is going to be based on their finances. And then also along those lines, Dr. Valentine, Medicare has capped costs now to $2,000 a year for all medical therapeutics. So that has put an upper limit on the Medicare patients for what they're going to have to expend.
This year it's 2,100, but yes, it is a wonderful plan. So, what other strategies have you all used in your practice to engage patients in discussions about using biosimilars to reduce hesitancy?
Well, as I mentioned in our practice, we prospectively send patients who are on the originator a message that they're likely to be switched to a biosimilar and give reassurance in that messaging.
Yeah, and I've been involved in the testing of these biosimilars, and I can tell you, as I describe the clinical trials to prospective patients, I would essentially describe these drugs as nearly identical, if not identical to the originators. And so I tried to reassure them that these were drugs that were already tested in the original form and that the biosimilar studies were a good option for them.
But again, the cost benefits are not to the individual patients, they're to society as a whole by lowering the overall costs of expensive medicines.
So, let's look at the resources available to help guide clinicians who are less familiar with biosimilars. FDA has published the Purple Book database of licensed biologic products. American College of Rheumatology has publications, Arthritis Foundation, and you can see here others that have published resources for professionals.
So we're going to try to take this all together now and sum up things. The first biosimilars that came out, or some of the first ones, were the TNF blockers, etanercept, adalimumab, and infliximab. These drugs were used for a wide variety of conditions, as we mentioned, inflammatory bowel disease, arthritis, skin diseases. And now we have lots of evidence that they're essentially the same, if not identical, in terms of efficacy and safety. We do think, therefore, that biosimilars are here to stay and should be viewed as a viable, safe, cost cutting alternative to branded biologics. So we want to reassure patients of this. We want to reassure doctors of this. Now, the rules regarding switching are often done on a state per state basis. There may be different rules in terms of the pharmacist or the payer actually inducing a switch over and above what the doctor is saying or writing in the prescription. So you need to know what the rules of your particular state are in terms of this being out of your hands as a prescriber and how much it's being done by a third party. And then the last kind of little issue is perhaps there will be situations, and I know this is the case in dermatology, where a prescriber of a biologic may want a particular class of drugs. For example, in psoriasis, we have IL-23 and IL-17 inhibitors, which are considered better than TNF blockers, but we don't have generics yet for those drugs. And there may be examples where you want to use a different class of drugs where there's no biosimilar, and then the pharmacist or the payer may be suggesting a biosimilar of a class of drug that you don't want to use. So there possibly could be some tension where you want to use the best drug and you may be forced, again, dependent on the payer, where to use a cheaper biosimilar.
And many of the societies, as Dr. Valentine mentioned, do have examples of authorization forms or reauthorization forms that can make it easier for the clinician to recommend the preferred mechanism of action rather than a specific product.
Right. So, thank you all for participating in this activity. Continue on to answer the questions that follow and complete the evaluation. Thank you so much to Dr. Blaubert and Hanover for your participation, and thank you to Medscape for supporting this program. Thank you.